I should be able to shift to this. And we should be recording at this point, so we're going to start on the lysosome with this. And then we'll try to go all the way through. So what have you heard about the lysosome in other classes? Have you heard of that one before? What does it do? Breaks down things. Some people refer to it scientifically as, well, it's a garbage disposal, right? So it's a part of the cell that's kind of a microenvironment that degrades molecules and cellular structures. It's has quite a few. It has a variety of different functions. And so let's skip this for a second, and we'll go through a little bit of the description, then we'll come back to that previous slide. So it can rid the cell of damaged, foreign, or unneeded molecules and organelles. And that's because the lumen, what does lumen mean? Yeah, an inner structure. So you can say the lumen of the gut, the lumen of the cell, the lumen of the lysosome. That means the inside of the lysosome has an acidic pH, approximately 4.8. Right, that's acidic. What's typical cellular pH? Typically pretty close to neutral, around 7, 7.4. What's blood pH?
It's slightly more basic.
Yeah.
7.4-ish. I don't know.
Exactly, no, you're spot on. 7.35 to 7.45 is blood pH. Very important it keeps within that range. If it drifts out of that range for a period of time, what happens? It's lethal. And what's one of the main reasons that it's so harmful? What happens?
Maybe like protons will like dissociate or reassociate into places they shouldn't.
Sure, that it can affect pH-dependent processes throughout the body and pH-dependent structures, especially proteins. Most proteins in our body are made to function at a neutral pH. As soon as that pH changes, protein structures can be affected, and proteins can denature. If there are significant amounts of denatured proteins, that can become fatal. And so our body has significant buffering systems to help maintain that blood pH. So if you ever hear anybody say, "Yeah, I'm on this cleanse and I'm changing my blood pH or my body pH, I'm becoming more alkaline and all this," I wouldn't do that. You can't, but you got to keep in mind that's misinformation. That's not something that a person can do or should try to do. When you eat alkaline foods, your stomach doesn't become more alkaline. There's certain things that our body resists and maintains a specific pH because that's what we have to live. So we'll talk more about that as we go through this. But one of the reasons it's acidic inside the lysosome to denature proteins and other molecules. So that's the point of it. Take proteins that are good at pH 7 and put them inside the lysosome, and those proteins denature. The combination of that denaturation and digestive enzymes can break down all those molecules. So inside the lysosome, there's about 36 digestive enzymes that break down proteins, polysaccharides, nucleic acids, and phospholipids, basically macromolecules, and any structures that are along with those macromolecules. So for example, a cell can target a mitochondria for the lysosome, and that mitochondria is completely degraded. Okay? So we'll talk more detail about that in a second. But what can you tell me about these digestive enzymes? Enzymes are typically made out of proteins. These are special proteins that can function at that pH 4.6. Just like the digestive enzyme in the stomach can function at a pH approximately 2. So there's some proteins that are made to function in specific parts of the body in a low pH. And that allows it to function while it's degrading the other molecules or proteins. So let's look at the next slide, and then I think we'll backtrack. That's probably the best way to do it. So here's the lysosome. Here's some specific catabolic lysosomal processes. For example, The lysosomes are used to remove cellular proteins that have been damaged or are no longer needed. Proteins, most proteins don't stay around for very long in the cell or in the body. Probably typical half-life of proteins, hours to days. So we'll make a protein, it'll stay around for a little bit of time, then it's degraded. Every time you exercise, for example, you stimulate the degradation of proteins within muscle fibers.
So you could like grow new ones?
Yeah, that's so basically it's adaptation. It's part of a dynamic environment and life that we have that allows us to adapt to certain things. If we made a protein that stayed around for 70 years, that protein's not really adaptable. Another reason is as a protein goes through its life, sometimes it's damaged, and the body will basically get rid of that protein and replace it with a completely functional protein. So it's really important for our health.
Do we recycle the amino acids that are broken down?
Typically, let me not say typically. Let me erase that. Oftentimes, yeah. Yeah, it's really interesting. When we talk about protein, we'll talk about nitrogen metabolism and how we go from protein to amino acids and then what happens. All right, another thing is phagocytosis. What does phage mean? To engulf or to eat, ingest, yeah. Cyto. That's cell. So for example, this is the engulfing of a cell. So the breakdown of foreign substances taken in from outside of the cell, such as used by the immune system. We've all heard of macrophage. What does that mean? It's macro. Macronutrients. Macro is large. Phage is basically a large cell that can consume other cells. So macrophages are made and they travel around so that they can recognize problematic cells or bacteria, engulf them, and then degrade them. So if you've seen pictures of macrophage or maybe little videos of them doing that, once they surround and engulf a cell, I think this one shows that. So rather than me just doing hand movements, yeah, this one. Phagocytosis. So let's say this is a macrophage and it recognizes this is a bacterial cell, bacterium. It can engulf that, incorporate it in, and then look what it does. It merges it with lysosomes. So now the macrophage uses its own lysosomes to degrade that particle or molecule. And then it's no longer harmful. Autophagy. This is an elimination of organelles from within the cell that have been targeted for breakdown. For example, mitochondria turnover rate in liver cells about 10 days. Mitochondria is made, functions for 10 days, degraded. And one of the reasons that's important is all those electrons flying around in that ETC don't always end up where they're supposed to. And loose electrons can damage other molecules. So a mitochondria is pretty good at maintaining its function and its integrity for about 10 days in liver cells. And then it starts becoming problematic and dysfunctional, and the cell has a way of recognizing that and breaking it down using its lysosomes. And then a better mitochondria replaces it. How about bone demineralization? Bone cells, for example, use lysosomes to degrade bone to release stored minerals into the blood. We've all heard of that, right? If a person doesn't consume adequate, consistent amounts of calcium in their diet, does it decrease blood calcium? Typically no. So that's not a good way to say, it's not good to say, let's see how much calcium you've been eating. We're going to do a blood calcium measurement. It's going to tend to be within a pretty strict concentration range. But if they haven't been eating calcium, what's keeping blood calcium at that level? Bones are being degraded. That's one of the mechanisms. And the reason is because there are things that have to have calcium that are more important in our body than bone. Like insulin release, like movement, like all different types of things that our body is willing to break down bone in order to maintain calcium levels for that function. So there are cells within bone that break down bone using lysosomes to put calcium into the blood. Remember that cell type? Osteoclasts are the ones that tend to degrade bone. So a good long-term indicator of calcium status, measure bone density, repeated measures. 329 lab, if you want, I'll everybody that wants to, I'll scan them, we'll get an idea of your bone density. Okay, so that's some important functions of mitochondria. I think this one kind of goes through that. And the last one we'll talk about over here. Later on in the quarter, we'll talk about this. Have you heard of this process? Receptor-mediated endocytosis. Sounds good, right? In other words, what's endocytosis? That's basically engulfing, in this case, a protein. So you're taking something into the cell by wrapping the cellular membrane around it. And it's specific because of a receptor. So a cell will put a receptor on the outside of its cell. When that receptor binds its ligand, whatever you want to call it, its target, then the cell can take it in. It forms this little endosome, intercellular vesicle, that can then fuse with a lysosome to separate out the proteins. We'll talk about this when we talk about heart disease and the LDL receptor and stuff like that. Okay, so this is receptor-mediated endocytosis. The lysosome is used to separate proteins. I think that's it for lysosomes. The nucleus. Great topic, lots of details. So now where we're going with this is we're going to go from the nucleus, its overall general structure, which you guys already know, and we're going to go to nutrient-regulated gene expression. Okay? And we're going to answer the question, why is vitamin D added to milk? We got calcium. Oh, really? How does that work? We're going to go through the steps so you can tell me how vitamin D regulates gene expression, how that affects calcium. Okay, that's probably one of the better examples, even though vitamin D is in FSN 329. Okay. The nucleus, largest of the cellular organelles. Nuclear envelope is double membraned. In other words, there's two membranes around the nucleus. It has pores or openings to allow passage of molecules. And that's a fantastic area of research. That's called protein trafficking. And the cell can basically let proteins through they're supposed to go through, and it can let proteins out. So it's kind of bidirectional, regulated transport through those pores. Remember, here's some terminology. DNA replication is the copying of DNA, and that has to take place before cell divides, right? We copy the DNA, and then both cells can have the genetic material that they need. And we said this before, transcription is the process of synthesizing RNA from a gene, and then the RNA can be translated. So basically we're changing the language from nucleic acids to amino acids, and that's how we end up with a protein. The presence of genes allows the nucleus to initiate and regulate most cellular activities. So we're going to talk about how it's regulated. So here's an important point to all of this. The DNA in all cells is basically identical. So in other words, a liver cell, if I isolate the liver cell DNA and I isolate the kidney cell DNA, they're basically the same. So why are they different cells? They have the same genetic material. Differential gene expression. Differential gene expression yields different proteins, and that allows cell types to be specialized. Why do we care? Because they have different metabolic pathways, different metabolic requirements, different fuel sources. So that's why we're paying attention to these details. Liver cells function differently than kidney cells, and they do different things. So we'll look at how that affects nutrition. And here's an example here. But remember Dolly the sheep? Boy, Dolly, that might have been before you guys were born at this point. What was the significance of that? Hey, we cloned a sheep. Yeah, what cell? A somatic cell. Not a germ cell. Not a cell that's normally involved in reproduction. In other words, you can take a skin cell. You can isolate the DNA from that skin cell. You can now take that DNA, put it into an egg, an oocyte, fertilize that, and you'll get a full organism from that. In other words, each cell in the body has all the genetic material that we need to make any cell in the body and make an entire multicellular organism. That was the most significant thing that came out of that. And here's why. The human genome, remember that's the term, all of the DNA in a cell contains billions of nucleotides. And you guys are living in post-Human Genome Project world. And what was the significance of that Human Genome Project? What'd they do? They sequenced every nucleotide in human DNA. So one of the first questions that came to my mind was, whose DNA is it? It's not mine, right? Everybody's a little bit different. Everybody's DNA in here, a little bit different. And that's why we look different. That's why we act different. That's why our nutrient requirements are slightly different. All of that. I might need more calcium than you do because of my genetic material. And so now what we try to do is we try to assign nutrient recommendations for everybody. Good luck. Right? Everybody's a little bit different. They act different. They do different things. So we're going to talk about some of the specific details of how that affects nutrient requirements. One of the things to keep in mind is only a small amount of these bases are part of genes. So let's say, for example, I have a chromosome that is a few million nucleotides long. And first of all, I have, how many billions of nucleotides in the human genome? Remember that? Approximately 4 billion. So 4,000 million nucleotides is the genetic sequence. And years ago, when I first kind of got into this, they used to say, yeah, but you know, a lot of it's not really that important. It's like junk DNA. Pretty egotistical for us to say that, because now we're like, oh yeah, you know what, it's not junk. It's involved in genetic regulation. And it's a structural component that determines lots of things, like which genes are expressed. So here's how it works, kind of. If we have all of this, let's say this is the double-stranded DNA of one chromosome, and let's say this is 10 million nucleotides. In other words, 10 million bases, AGTC, right, throughout. Here's a gene. Here's a gene. Here's a gene. Small amount of all of those nucleotides are involved in genes. So the question is, how does the cell know where a gene is? Proteins find them. So it's all regulated. So how are the genes identified and expressed? Proteins, called transcription factors, remember that term? Any protein that's involved in transcription in any way is a transcription factor. Nutrients regulate some transcription factors. And that's how they regulate gene expression. So these proteins find specific binding sequences. They bind that sequence, and then transcription can be initiated. I loaded a video on Polylearn that shows transcription. It shows the assembly of the transcription complex and the actual synthesis of an RNA molecule. If you haven't seen that before, you might want to look at that again. It's pretty cool. So here's some more terminology. Promoters are regions of DNA. So in your notes, put promoters for us are nouns. Promoter is a noun. Because they're not promoting something, it's being promoted. We'll go through that when we talk about this. Right? Promoter regions are genes, are regions of DNA that are typically upstream or in front of genes that are targets for proteins and enzyme binding that promote the expression of genes. So proteins bind promoters. And that process can promote the expression of a gene. Went through this. So transcription, translation, we said overall this is gene expression. Because now if we express a gene, we can get a protein from that gene. Remember the DNA is unique to each person? Because of that, proteins tend to be slightly different. Remember there's a difference between DNA and RNA, so a lot of this information's in your chapter 1, so please read through that. And this is really important for a few reasons, not only this quarter but next quarter. 329, hopefully you'll talk about RNA structure, because that's an important part of nutritional science research. So here's DNA on the left, RNA on the right. Remember this little difference right here? It's deoxy, right? There's not a hydroxyl group here. That's one of the reasons DNA is more stable than RNA. Remember, RNA is susceptible to a nucleophilic attack because of that hydroxyl group. So just remember, DNA is more stable than RNA. We know that. We've all seen Jurassic Park. Right? I have a story about that. I won't tell you today, though. But this is one of the other things that's unique. So that's one unique difference. You want to have in your notes unique differences between DNA and RNA. Lack of that hydroxyl group on the ribose. Another thing, DNA is double-stranded. RNA strands are single-stranded. And even though they can fold on and form secondary, tertiary structures, they are single-stranded by nature. So we go from 2 strands to 1. Another difference, DNA has thymine as one of its nucleotides. RNA becomes uracil. But of course the other ones are adenine, guanine, cytosine. A little bit about the genetic code, which I think is again, I'm going through this because it'll involve my example of vitamin D in milk. Okay? It's more interesting when your genetics involve a glass of or something we eat, right? So here is a DNA molecule. Has one strand that has a direction of 5' to 3'. Remember that? And the direction is basically determined by the presence of the open hydroxyl group on the nucleotide. So I won't go into that detail, but here's the sequence. And remember, one of the strands is called the sense strand. It's called the sense strand because this sequence is used to determine the amino acid sequence of a protein. That's why they call it the sense strand. Sense strand determines the amino acid sequence of a protein. The antisense strand is complementary. Because of that, it basically runs backwards, all right? Remember that? So when you're first doing this and you have a piece of DNA and you're trying to find genes, your software will actually look for open reading frame, sense, antistrand, sense, all that stuff. So now, for example, if you go from DNA to RNA and you make a complementary copy of the antisense, you're essentially copying the sequence of the sense. All right? So if we go ATG here, and now I go, okay, this is complementary. In other words, A binds T here, G and C pair, right? So in other words, if I go ATG here, it's TAC here. It's complementary, which means these sequences. Now, if I copy this, if I make a complementary copy of the antisense, I'm copying the sense. Right? So now I got a T here, and I go, okay, that binds with an A. And now here's my A. I'm going to go T, but because I'm making RNA, that's a U. And now I go, okay, now what's complementary to C? G. So there's my G. Remember that? Okay? So now I have my mRNA, which when it's translated, changes to amino acids, right? So each one of these, remember, are triplet nucleotides that make codons. That codon is recognized by a specific tRNA, and that incorporates the appropriate amino acid into the sequence. Remember all that? You've seen it a few times, but so now I know the proteins always start with methionine. So this is my initiating codon here. And then from then on, it's read 3 nucleotides at a time. Once we get that in frame, we know. Now, when this mRNA goes through a ribosome, those tRNAs are binding, and you're getting 1 amino acid per 3 nucleotides. So here's the genetic code. And if the first base is, for example, let's look down here, and we say if the first base is a G, and the second base of that codon is a U, and then the third one is a G, a tRNA loaded with valine is incorporated into the protein sequence. But remember, there's redundancy in the genetic code. So let's look. Let's look right here, for example. Let's say in one of my proteins, the nucleotides read GCG. So in my protein, I get an alanine there. I don't know if you remember the structure of alanine. It's just a methyl group in a side chain. It's pretty innocuous. It doesn't affect protein structure a lot. Okay. Let's say in all you guys, rather than GCG, it's GGG. That codes for glycine. Glycine is also a very common, I wouldn't say non-effective, but it doesn't generally affect protein structure significantly. It's just an H in the side group. So that's the simplest one. Okay? So in other words, we can have genetic differences that don't change the protein structure. Or let's say mine is GCG and you guys are GCA. It's the same amino acid. So sometimes a nucleotide change between people changes the amino acid. Sometimes it goes from one amino acid to another, but the characteristics of the amino acids are not significantly different, so protein structure is not affected. However, let's say, for example, in this other case, we go from this. We go GCG to GAG. That would cause glutamate. Remember glutamate structure? It's charged. So now we have a charged amino acid. There's a chance that could change protein structure. If this protein has to do with calcium metabolism, it might mean that my calcium requirements are different than yours. And I'll show you how all that works. And that's one of the reasons, right, we've all heard of personalized nutrition. Hey, you know what? We've got the Human Genome Project. We can have personalized nutrition. We're a long way from that. I think the next step is trying to assess differences between people using traditional markers like inflammation markers and risk of heart disease and stuff like that. When you start getting into the genetics, there's a lot of factors that we are still working through. So it's going that route, but we're not quite ready, I think, to look at genetics and basically tell a person everything they should eat. But that's kind of what they mean by that. And this is how it works. It's all very sequence specific. And this is how. So if we look at this structure, this shows the DNA at the bottom, obviously, and a protein that's actually a dimer. So this is a homodimer where 2 proteins are bound together and then they can interact with the DNA. The protein-DNA interactions depend on bonds between specific amino acids of the proteins and nucleotide bases. So in your notes, you want to say, the protein-DNA interactions are typically, what kind of bonds? Hydrogen bonds. And the interactions are sequence specific for nucleotides and amino acids. In other words, let's say this nucleotide right here, which is going to interact with this part of the protein. So this particular nucleotide is going to interact with a specific amino acid of the protein. If my nucleotide is different from everybody else's, it could affect this interaction, which could affect protein binding. But if my protein sequence is different than yours and this amino acid is different, that can also affect this interaction. So the point of it is the protein DNA interactions are affected by sequences of both the protein and the nucleotides.
What's a common example of a protein-DNA interaction?
We'll go through that in a second, but like transcription factors, we'll talk about tau-binding protein, we'll talk about nuclear receptors. Yeah, so good question, that's where we're going. Milk, calcium, right? Okay, so a change in the protein's shape can alter the protein-DNA interactions, and some proteins rely on nutrients to change their structure and shape and allow them to bind DNA. So for example, vitamin D binds a protein, changes that protein structure, that protein can now bind DNA. So that's where we're going. Here's a map of a gene, and in here we're going to talk about eukaryotic mammalian genes. There are some differences between us and prokaryotes, a lot of them. Have you ever seen these? Remember these? This is like a map of a gene. So let's define some of these. What's +1? Transcription start site. Right, so here's DNA, double-stranded. We've got some rectangles here, different colors, and we've got an arrow +1. That indicates transcription start site, which means that's the first nucleotide of the RNA. Remember, transcription is DNA to RNA. So this is where transcription starts. Okay? There are other things that regulate how often and how quickly transcription starts. Okay? So when we look at these little boxes here, these little, is that brown? Orange, yep. The orange boxes here are defined here. This is promoter, proximal element. And in your notes right now, put an element equals a short nucleotide sequence. An element is a short nucleotide sequence. Right? And look where they are. Upstream of transcription start site. All right, we know it's upstream. See that arrow? We know transcription is going to go this way. So remember this? There are proteins that are going to bind right here. And they're going to bind, the proteins are going to use their amino acids to bind specific nucleotides. So you can do this in the lab. We used to call this promoter bashing, where you take a specific sequence of DNA that you believe regulates the expression of a gene, and then you start looking for footprints of transcription factors. In other words, you can use assays, so I'm sure it's a lot easier now than it used to be. You can find out exactly where proteins are binding to the DNA. And that's what those rectangles are, those little boxes. That's where protein binds. Protein binds here, 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 and here. So those are protein binding sites within the promoter. So this area right here is the promoter region. I really don't have that here, do I? So you want to define a promoter as a part of a DNA sequence that regulates the expression of a gene. The promoter region is a part of a DNA sequence that regulates the expression of a gene. Expression of a gene. So in other words, it regulates transcription, right? Is this all really basic review?
That's kind of like left, right?
Yeah, that'll whack out, yep. Except,
That's just like an example.
yeah, and they're simpler in a lot of ways. So there'll be some differences coming up between bacteria and us that are important. Okay, right, you got that? So now imagine, here's where we're going. There. This is what we got to do. Here's a promoter. Here's all the proteins they're going to bind. So we're talking about how we go from that DNA to this so the transcription can occur. Okay? What else? See the way it says proximal, promoter proximal? In other words, close to the start site. And we're pretty close. The minus 200 is minus 200 what? Nucleotides, base pairs. Yep. All right. Here's remember, you can cross this out. See enhancer, yeast, UAS. You can get rid of that. Let's talk about people. What's Tatabox? Remember that? What binds that? What binds Tatabox? Tata binding protein. Remember that? Tata binding protein binds a Tatabox. So we'll look at that as actually,
Will you ask us to draw anything like this to this level or to the level that it is in the next few slides? You like had another one that was a little bit more in depth. Not that one.
yeah, that's where we're going. You'll add proteins that look something like this. Guys, we're not going to do anything in here that I know you can't do. And the reason this is important is because some of these proteins are regulated by nutrients. And so we need to talk about the basics of transcription and then how it's regulated by nutrients. And we're going to do that all quarter this quarter, and you're going to do it in 329 as well. This is how things that we eat affect gene expression. It's also about heart disease. It's about cholesterol levels in the blood, things like that involve this. So as we go through this, this is something you want to know very, very well. Okay? We're almost ready to put all this together. What else? Oh, let's go this way. Exon, intron, exon. What's an exon? Yeah, so that's a nucleotide sequence that determines amino acid sequence. So we say it's expressed in the protein. So exons determine amino acid sequence. Introns are intervening sequences that do not affect amino acid sequence. Remember that? Yeah, they're spliced out. Yep, they're pulled out. Okay, so we'll talk about that in a second. But now here is, we'll come back to this later. But you can see VDR, I'll give you a hint right now, that stands for vitamin D receptor. So this is a protein that interacts with, see it right there? That says D3. That's a form of vitamin D. So this is what happens. Vitamin D goes into a cell, binds to a protein inside the nucleus, and regulates gene expression. Okay? So we're building out to that. Right now, let's talk about basic transcription. So what happens is, and I'll give this to you now, and I'll kind of give you a little big picture now, and then probably next time we'll go through the specific steps of this process. And you guys, on quiz 1, will definitely have gene expression. We probably won't have protein secretory pathway, but definitely gene expression. So go through this in detail. So what's going to happen is, I want you guys to give me the steps in gene expression with a diagram. Okay? So that's what's coming. But let's look at some of the things that happen here. So here's the Tata box. And the Tata box, they call it that because it's basically a repeat of about 9 to 15 nucleotides that look like TA, 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 TA. You ever seen that sequence? I don't know if you remember that. That nucleotide sequence is bound by Tata binding protein. That's what this guy is. So Tata binding protein binds Tata box. And that's why I put this up here. This is a Tata binding protein. And it will sit right in there looking for its specific sequence that it's supposed to find. In eukaryotes, more than half the time, Tata binding protein is the first protein there. Okay? So I'll go through this next time. Tata binding protein, when we said, how does a cell know that that's a gene? One of the main reasons Tata binding protein is looking for it. And once it finds its TA repeating sequence, it'll scan the DNA and go, oh, binding right there. And then guess what it does? It signals other proteins. Okay? So that's what this means. Tata binding protein finds its Tata box and binds, and then look what it does. It starts recruiting other proteins. I'll use that term a lot. This is transcription factor recruitment. In other words, one protein is bringing in other proteins. And so that's what is happening here. Now you have another protein that binds. This is transcription factor 2B. That name is not important for us in this class. Now, so this is an auxiliary factor. So this is another transcription factor. And they eventually bring in this guy. What does that stand for? POL2. That's a polymerase. And for us, transcription for proteins is done by RNA polymerase 2. So in your notes, RNA polymerase 2 does transcription that yields proteins. Obviously, there's an RNA polymerase 1. There's also an RNA polymerase 3. One of them makes tRNA, the other one makes ribosomal RNA. That's not important. We're not going to go into detail on those, but remember, RNA polymerase 2. Prokaryotes have one RNA polymerase. So right under your notes where you said RNA polymerase 2, put RNA polymerase 2 does not recognize specific DNA sequences. RNA polymerase 2 does not recognize specific DNA sequences. It relies on other proteins to do that. What's an example of another protein that it probably relies on? Tata binding protein. Yep. So you could put, for example, Tata binding protein. So in your notes, you can then put Tata binding protein is required for the expression of more than half of eukaryotic genes. Sure, Tata binding protein is required for the expression of more than half of eukaryotic genes. So it's often the first one there that then recruits other proteins that eventually recruit RNA polymerase. So let's kind of go out big picture before we go. One more comment. This is a little bit complicated for transcription, and the reason it's complicated is because there's lots of proteins involved. But that also increases the possible options for regulating transcription for us. Prokaryotes will go along and they'll go, hey, as long as we got this, we can grow. A lot of our genes are regulated by a lot of different proteins and factors, and that's why it's kind of complicated. So we're going to build a basic transcription complex, and then we're going to talk about how vitamin D regulates that. And then that'll give us a good basis for other things that we'll talk about through the 2 quarters. So we're getting there. We almost got to the vitamin D part. Uh-huh.
So I wrote down that RNA polymerase II does transcription that yields proteins, but this is all a part of like transcription, not translation.
Correct.
So it's these proteins are not the result of translation; they're different.
Sure, they're the result of translation, but RNA polymerase 2 makes mRNA that can then be translated to a protein. RNA polymerase 1 and 3, they don't make mRNA, they make tRNA or ribosomal RNA. They don't determine protein sequences. Yep. Okay, guys, any other questions? Okay, guys, thank you. So I'll see how I do with this video, and I'm going to stop it now, and then I'll see you guys Thursday.
For quiz 1, what would be your preferred study method?